The first thing we need to determine is the gene that will bring the desired trait to the crop plants. The next step is isolate and amplify genes of interest from the donor organism. In the case of Roundup Ready or crop plants resistant to the herbicide glyphosate, the gene of interest was derived from the common Zoobacterium agrobacterium strain CP4 that encodes for an ESPS enzyme, or from Orchobacterium anthropi that encodes for an glyphosate oxidase enzyme. Both have high tolerance to glyphosate, so the transgenic canola plant can keep producing aromatic amino acids. The polymerase chain reaction allows these genes to be cloned into multiple copies. Once the genes have been multiplied, scientists design the genes to work inside a different organism by cutting the genes apart from the enzyme. The transfer of the gene is the next step in GM process. Tissue culture is used to spread masses of undifferentiated plant cells called collars where new transgenes are added. The new gene is inserted into some of the cell using different technologies. The most common methods include gene gun, agrobacterium, microfibers, and electroporation. The main goal of each of these techniques is to deliver the gene into the nucleus of a cell alive. One example, the gene gun can be used on tissue culture cell of any species. It works by shooting DNA, which are coated on a microscopic gold particle, into the plant cells. The gene gun places the tissue culture cells in a vacuum chamber and then propels the metal particle under a high-pressure gas. Transformed plant cells are then regenerated into transgenic